In this video, we will continue our discussion about effect of isoferulic acid on pancreatic cancer cell lines. In the previous video, we have seen some of the evidence that with isoferulic treatment in the pancreatic cell lines and BXPC3 cell lines, we see a dose-dependent increase in the apoptotic signals, and we have checked the results using tunnel assay. Then also we check the results using comet assay. So in this video we will discuss the result. We will discuss what is actually happening within the pancreatic cancer cell lines that cause this apoptosis. So one of the hypotheses that researchers have is that there could be intrinsic apoptosis might be triggered. In order to check that hypothesis, they check the mitochondrial membrane of the pancreatic cancer cell lines, as the intrinsic apoptosis begins in the mitochondrial membrane. So in order to check the healthiness of the mitochondrial membrane, they use JC1 aggregates. So when the mitochondrial membrane is healthy, the JC1 monomer aggregates within the mitochondria, and if mitochondrial membrane is not healthy, then JC1 monomers aggregate less in the mitochondria. So how we can identify whether the mitochondrial membrane is healthy or not? With the ratio of JC1 aggregates by JC1 monomer. If it is high, then mitochondrial membrane is healthy. If it is low, the mitochondrial membrane is unhealthy. And since high and low are relative terms, so we will compare the results with the control. So in the control for the Panc1 cells, we can see that it has some monomers which are shown here, and aggregates are red fluorescence in nature, and the monomers are green fluorescence. So these are JC1 monomers are designed in that way. So first, in the case of control, we can see the around 6.35% monomers are there, whereas maximum of them are these aggregates. But when we treat them with isoferulic acid, we see there is an increase in the number of monomer. With the low treatment, it increases from 6.35 to 11.34, and with medium treatment of isoferulic acid, it increased to 24.16, and thereafter it increased to 38.61. So this indicates that isoferulic acids will affect the mitochondrial membrane that might trigger apoptosis. And the quantifications are shown on the right hand side. Here we can see that the JC1 percentage monomers are increasing with a dose dependent increase in the isoferulic acid. And similar things they have seen for BXPC3 cell lines, that with the increase in the isoferulic acid treatment, they see an increase in the monomers. And here also we can see that it follows the same trend, that is with dose dependent increase in the isoferulic acid treatment, there is an increase in the JC1 monomer. So once they identified that it's the mitochondrial membrane that got damaged when the pancreatic cancer cell lines are treated with isoferulic acid, they wanted to check whether it will trigger this pro-apoptotic factors or not. So they checked for this cleaved PARP and cleaved caspase 3, which are the markers for the apoptosis, which are the markers for the apoptosis. So they can see that with increase in the dose dependent concentrations, with increase in the dose dependent concentration of isoferulic acid, there is an increase in these apoptotic markers, that is cleaved PARP and cleaved caspase 3, both for the Panc1 cell lines and also for the BXPC3 cell lines. Apart from this apoptotic factors, there is one more phenomena that occurs within the intrinsic apoptosis, that is cytochrome C migrates from mitochondria into the cytoplasm. They wanted to check the amount of cytochrome C in the mitochondria and in the cytoplasm. So when they check this cytochrome C using Western blot, they are able to see that cytochrome C decreases in the mitochondria, whereas it increases in the cytoplasm with dose dependent increase in the isoferulic acid treatment. And similar trends they have observed for the BXPC3 also, that is cytochrome C goes down in the mitochondria with a dose dependent increase in the concentration of isoferulic acid, whereas it increase in the cytoplasm with a dose dependent increase in the concentration of isoferulic acid. And for controls in the mitochondria they use COX-4, and for the cytoplasm they use beta-actin, which was signals remains almost constant in all the cases. So this indicates that is the cytochrome C phenomena seems to be in line with the cells are under in line with a condition where cells are under apoptosis, or where apoptosis is triggered within the cells, or intrinsic apoptosis more precisely. So I hope you understood how they validate their hypothesis about intrinsic apoptosis. First they check the mitochondrial membrane healthiness, and thereafter they check the apoptotic factors and the cytochrome C migrations from mitochondria to cytoplasm. So if you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.